Hello, it's Mrs. Murdock, and we are doing a review session for AP Biology for the Unit 2 test on cells and cell transport. So the first thing I thought I would do is just very quickly go over the basic topics and terms that you should be studying. All right. I'm not going to go into great detail here, but obviously the first big topic is cell structure and function, right? So within that, you need to know prokaryote versus eukaryote, right? The structure of a prokaryote versus the structure of a eukaryote. Okay? So if I were going to do a really quick, super fast review of this, I would remind you that that means that we have here we have bacteria, and that is it. Okay? We have bacteria, we have small and simple, and these are all unicellular, right? Okay. If we're talking about comparing that, there are a lot of other things we could say, but I'm doing this quickly, okay? Versus eukaryotes, the kingdoms would include, please remember, that includes protistans. Protista, fungi, animals and plants. Those are all kingdoms of life that are made up of eukaryotic cells. They are larger cells, right? Larger, complex, bless you, and lots of membrane-bound organelles and compartmentalization that makes them able to be large and makes them able to do chemical things that the bacteria can't, right? And here's where I want to get at a misconception that I keep hearing you say. Protista, some are unicellular. There are unicellular eukaryotes. Please remember that your slime mold is included in this. It is a single-celled protistan, and therefore it does not divide by binary fission. About 10 of you told me this in your essay, and this is incorrect. Okay, so remember that some are unicellular, and anything that's a eukaryote is going to divide by mitosis. And I know we haven't gotten there yet, but that was such an egregious mistake. I wanted to make sure I addressed it, all right? Most are multicellular, right? Most are multicellular. The fungi, the animals, the plants, they are by definition multicellular. Protistans, though, there's a hell of a lot of them that are single-celled, right? So, just so that you understand that just because something is a prokaryote versus a eukaryote, the eukaryotes can be unicellular too. Just remember that. Our slime mold was an example of that. Okay. All right, going on. So, you need to know particularly eukaryotic organelle structure and function. And that has to go beyond what you learned in freshman year, right? So remembering that the mitochondria is the powerhouse of the cell is great. That's fine. But you got to know more than that, okay? You should be associating mitochondria with ATP molecules. You should be able to do that. We learned that in biochemistry. You should be making that connection, okay? So you need to make sure that if you haven't looked at your eukaryotic organelle structure and function in a while, if you have that guided reading, go back and look at it, because there are details there that you will need to know. All right, the endomembrane system. The endomembrane system. Right, so remember that this is a really prime example of when organelles work together. So this is really more about, how do you get proteins to get made and then processed and then moved to where they need to go. That's what the endomembrane system is about in a nutshell, right? And depending on what type of protein it is, it might take a different pathway. So be really careful about this, okay? I mean, basically, we're starting with the ribosomes, and then going and going and going until you get either to the membrane, right, to be secreted into the outside of the cell, or you might wind up inside a lysosome because you're a digestive enzyme, and that's where you're going to be, and you're going to stay inside the cell. Or you might even go a totally different way and not really go in the endomembrane system at all and become a cytoplasmic protein. It's kind of small, sorry about that. I'm going to raise this up so you can see it a little better. There, okay. So remember that ribosomes, you should remember, are where proteins are made. That's where they're put together. And then the fate of those proteins depends, where it goes through the endomembrane system is going to depend on what kind of protein it is. If it's going to stay in the cytoplasm, it's not going to be in that endomembrane system at all. It's going to, the RNA is going to just go from a cytoplasmic ribosome, and it doesn't need to even go to the Golgi body at all. Why would it? But if it's going to be secreted out, then it's going to be, in the ER, and then go to the Golgi body, and then vesicles are going to take it over and bump it up against the cell membrane and spit it out into the environment, right? So that might be different, though, if it stays inside a I didn't spell that right. Look at that. You should have told me. Lysosome. Come on. Lysosome. Right. If it winds up in there, then it's going to butt off the Golgi body and not go to the cell membrane and just hang out in the cytoplasm, because that's where it's going to go. So this is the level of thinking that you need to have, right? So when it asks you a question about the endomembrane system pathway, you need to make sure you know what kind of protein is it asking about. Really read the questions. Okay. On to, let's say, after endomembrane system, you also know that we talked a lot about surface area to volume ratios, right? All right. I'm not going to go too deeply into that, and consequences, right? So that is something that you know how to calculate, hopefully, right? So if they give you the surface area of something and then they give you the volume of that something, you can calculate the ratio. And if they ask you to compare two different cells to each other and they give you the surface area to volume of those cells, you can do that calculation and then compare. That might require you to organize yourself a little bit on the paper. And since these tests are on paper, make a little table for yourself so you can do that calculation. I'll show you that on one of the progress check questions. All right. Endosymbiosis is another topic you need to know. That is the one topic in this unit that connects with evolution, right? So endosymbiosis is really talking about how did eukaryotic cells evolve in the first place. About 2 billion years ago, if you look at that painting up there, a long time ago when the conditions were right, there was a larger prokaryote that ate a smaller one and didn't digest it. But there's a little bit more to it than that. I haven't really emphasized that, but if you read about endosymbiosis, they talked about, yes, that's the origin of the mitochondria, yes, that's the origin of the chloroplasts, it's also the origin of whatever organelles you have in there that are double membraned, including a nucleus, including some protistans that have not even double membrane, but sometimes triple membrane organelles in them. That's evidence of phagocytosis, right? Putting a single membrane around something, so now it has two. And then phagocytosis may be happening again to create a second or a third membrane around it, right? So that is evidence of endosymbiosis, having multiple membranes around organelles, because each new membrane came from a phagocytosis event that happened 2 billion years ago, and then digestion didn't happen, right? So it's not just as simple as mitochondria and chloroplasts. It's anything in there that has lots of membranes. Okay. Cell membrane structure and function, right? cell membrane structure. And I do mean cell membrane. So remember this picture, right? Oops, I'll try to make that neat. My phospholipid bilayer, yes? And if you just think of that as the basis, you know that there's more to the cell membrane than that. You've got proteins embedded in there, you have cholesterol embedded in there, but just this part of it, where is the hydrophobic part of this structure? Right here, right. So you need to know where your hydrophobic or nonpolar parts are, right? So you know that that's all hydrophobic and that the heads out here are hydrophilic at both ends. Right? Hydrophilic. Make sure you know those words really well and you know where they go when they show you a molecular structure, because they're going to do that. Okay? Cell membrane versus, I'm going to actually add something here, cell wall, because I'm seeing a lot of confusion with these two things. Cell membrane and cell wall, very different structures, very different purposes. So with the cell wall, you're talking about usually you're talking about a rigid structure made of some sort of a fiber. And it is almost always completely permeable, right? Versus the cell membrane structure, which is semi-permeable. Remember that? There's some words there that people have a tendency to stumble over. Okay. So these are very, very different structures. Remember that a plant cell has a cell wall on the outside of itself, again, completely permeable, only there for rigidity and structure, made of cellulose. And then inside it, it has a thinner cell membrane. I'm trying to draw it so it looks like it's separate, right? So cell wall is here for rigidity on the outside. Cell membrane is the inside line there. Completely different functions, completely different molecular structure. Okay. All right. Cell transport is the second part of this, and I'm probably going to run out of time here before I want to, but okay. So there's cell structure and function. Then we have cell transport. Cell transport, I think for a lot of you, was more challenging than remembering all those cell parts and remembering what they do and all of that, right? Because cell transport involved passive versus active, right? Versus active transport. And if we make a little T-chart here, right? Here we have diffusion and anything that doesn't require energy, right? We have diffusion, we have osmosis. We have facilitated diffusion. If there's a channel that needs to help the molecule, facilitated diffusion. Okay, all right. And over here we have anything that is a pump or bulk transport, that where part of the cell membrane literally has to contort itself. That takes a lot of energy, right? So passive, no energy. Active, yes energy. And passive is going with the gradient, right? And active is going against the gradient. Okay, I only have a few more seconds left here. So cell transport tended to be the bigger thing that was difficult. So predicting water flow was the main thing. Predicting water flow, or predicting which side of a U-shaped tube would go up or down and why, is the biggest thing there. And then along with that was being able to figure out how kidneys work, right? Kidneys function with nephrons. We spent some time on that. And how nerves function within neurons. Okay, and these